What we're looking at is ways in which we can identify individuals who are at higher or lower risk of breast cancer. And so what I was covering was genetic markers that are associated with breast cancer risk. And there are kind of two types. There are some rare variants which are associated with a high risk of breast cancer, and there are other more common genetic markers which are associated with more moderate risk of the disease, and we've identified over 100 of these markers. So what we're looking at is ways to combine these markers together into a risk prediction score. And so the idea is that if you combine the information from all these markers together, you can identify individuals, women who are at higher or lower risk of the disease. And we've had this strategy for a number of years with some known high-risk genes like BRCA1 and BRCA2. But what we have now is a much larger array of markers that we can use, and that enables us to identify a much bigger group of women who are at significantly high risk of the disease or significantly lower risk of the disease. And so now we can think of ways in which we might intervene by using more intensive screening, for example, or chemo prevention for the higher risk women. There are a number of challenges. I mean, the first challenge is understanding how to combine the information to define a risk for an individual. And for that, we need some better tools. We have the information, but we need some ways of making that information available to clinicians at every level, from primary care to specialist clinics, so that they can interpret the information well. And I think there are a number of other challenges then in terms of, because we'll be identifying potentially a lot more high-risk women, there'll be the issues of coping with the demand. So, for example, one of the interventions that is generally considered appropriate for high-risk women is screening with MRI. If we identify a lot more women who would be eligible for MRI screening, that has some implications in terms of resources, in terms of the number of available machines, for example. What we've been able to do is to define the proportion of individuals, the proportion of women who are at different levels of risk. And we can, for example, identify about 4% of women who are at a greater than threefold risk of breast cancer. And so in many countries that would be considered high risk, the sort of the women who would be eligible for more intensive screening or potentially even prophylactic surgery. And that 4% of women, they explain about a fifth of all breast cancer cases. So it's potentially quite a cost-effective strategy in terms of improving breast cancer prevention.